Okay, good morning. Let's have a class. And today's lab follows the fact sheets will be chapter 3, smear preparation and simple stem. So again, we're going to give you about 15 minutes opening remarks and then show you how to do the simple stem, then you do it. So lab 2, we will talk about simple stem. So first thing first, why we can do staining for bacteria. Most of the time, the bacteria grow at 35 degrees Celsius for 24 hours. Then, what are you going to see it? Most likely, it is negatively charged. So, negative charged. And we have lots of dye, which is a chemical ingredients. And right here, you could see some of that. Let's say methylene blue, the blue one, and you have saffronin. And lots of you will see a big purple one, which is crystal violet, which is on you dye rack, you see the purple one, which is crystal violet. So we have those chemical dye. Such as, I use different color, crystal violet. It's a purple one, saffronin, red to pink, and we have a blue one, which is methylene blue. And sometimes we have a green one, which is malachite green, for example. Malachite green. Most of these dye, or all of these dye, which is actually positively charged. Therefore, they could combine with each other and stick together and will showing some kind of the color. Okay, so the background is like bright, and you do the stem, you do the observation, it will be showing kind of the color. Here I want to mention that crystal violet, saffronin, malachite green, the carbon fusion, all those are very good positive charged dye, except for methylene blue, a little bit weak. So usually, as a learner, you don't have a much staining experiences. We usually don't choose methylene blue, but we choose others to do a simple stain. Now, why we wanted to do the simple stain? Then the second question. Because we want to see the morphology of bacteria. And I would say a bacteria cells, morphology of bacterial cells. So what are they? That could be these like a dots. We call it a Koch cell. It could be like a line, okay? We call it a logs. It could be a little bit twisted circle. We call it a vibro. Or we call it a spiral. So these are some of the basic morphology. So morphology actually means shape of bacteria. It's just to change a new terminology, but it's the same thing. We also want to do the simple stem because we want to know the arrangement of the bacterial cell. So what are the examples of the arrangement? It could be just a single cox cell, a single rods. So we will say single cox cell, rods. It could be two of the cox cells stacked together. We call it a diplococcel. Diplococcel. It could be some of the cox cell looks like rods, but it's still cox cell. So we call it a coccobacillus. And it also could be a four cox cell stacked together, like these, like that. This is, we call it a tetrad. It also could be, looks like a grape, a bunch of cluster. So we call it a grape cluster. This is typically for Staphylococcus aureus. So this is arrangement. What we want to see is underneath the microscope. The reason is some of these morphology and arrangement will be very typical for a pathogen. So for example, if somebody have a cough, have a sneezing, have a fever, then you get a sputum sample, you do a simple stain, you find it's a diplococcel, possibly Streptococcus pneumoniae. And you have a kids who had a lethargy, a very sleepy, and losing like a focus, and have a fever. If he didn't have a vaccination, it could be Haemophilus influenza, then it could be a coccobacillus, looks like. And sometimes you get contaminated with E. coli, O157H7. So you do a stain, you find it's a single rod just. So that's why it is important, because these arrangements, these are morphology directly related to some of the pathogen, and we know the symptoms, we could get a very preliminary diagnosis, and we could further do some of the treatments. And obviously could use antibiotics sometimes if it's needed. So this is a very basic story behind the simple stain. Now next topic, since we are in the lab, what we're going to do the simple stain, so will be the procedure. Okay, for the simple stain, we need first of all is a glass slide. So number one, we need a glass slide. You should have a glass slides in a box in your personal basket. You may need to clean a little bit, okay? Use alcohol, use just general water, paper towel to clean it. So we're gonna do a simple clean. And very important, we're gonna have to do a label. How we do it? You draw a big circle in the center. And today we will do an E. coli simple stain. So we just write EC on the side, represents E. coli. Okay, the second step, flip over glass slide. You have to flip it over. Why? You put the Sharpie marker there. These marker sometimes will be damage the lens of a microscope. So make sure you flip it over. Especially in the old time, sometimes in those satellite lab, they use crayon to do the label. Then the crayon definitely will damage the lens. So flip it over. Okay, now what are you going to do? At this moment, you need using a loop. Let's say we already labeled. You need to use a loop. This loop is sterilized. Sterilized, which means you're going to back and forth to do the burning on the Bunsen burner. And then pick some of the water, one drop of water right on the middle. Okay? Then what you do? Sterilize the loop again, and you're going to pick a bacteria. Today you will pick a bacteria from a plate. So we will pick is some of the colony right here. So use loop to pick a colony. You have a water here, and you have a bacteria here. You're going to make a big smear. This is going to make a big but thin smear. Don't make a thick one, because we want to see the morphology and the arrangement. If you make a thick one, you will see a big cluster. You're not going to see each individual cell. Okay, so make a smear. After doing this, it's very important step. Air dry for 3 to 5 minutes. Let it be completely dry in the air. Why? If you do not do the air dry, later on you put the slides underneath microscope, what it looks like, you see it's a lot of legs. It's not going to be clear. So completely air dry, 3 to 5 minutes. Then the step is called heat fixing. What means the heat fixing? You had a Bunsen burner right here. You have a fire, and your glass slides will be on the top. Of course, use faucets to back and forth a couple of times. To use heat to let the bacteria stick on the surface, because at this moment we are cooling the bacteria enzyme. That's called heat fixing. Now we move on to the stage is move this glass slides to the sink. Of course, you're going to put on the rack, okay? So let's say this is a sink, and then we have all these racks, we put it there. Now, we're going to put a smear right here. We're going to put a dye. What other dye we're going to using? It's right here. Crystal violet, saffroning, malachite green, carbon fusion. Let's say we put crystal violet there. So put one big drop of crystal violet. We will staining for 1 minute. What is 1 minute? 60 seconds. You could use timer to do, or you can just count in Mississippi for 60 times. It's the easiest way to do. Okay. Next step. After staining for 1 minute, we will be gentle rinse for about like 30 seconds. Okay, just a very gentle rinse. And last step, dry, and then underneath the microscope, we use 10x to find specimen, which means a sample, and then you transfer to 100x with oil immersion. We will go to do the observation. And today, what we're going to record on your notebook is a morphology and arrangement of bacteria. So is it a large, coxal, vibrio, spiral? And what's the arrangement? Is that a single cell? Is that a diplococcel? A coccobacillus, a tetrad, or it's a grape shape? So these are the things what we're going to do for the symposium. Okay. Next, same as before, we will give you, and I will do an example for you how we do it. This time, I will be using this stage as an example. So everybody, just come right here. We're going to show you how to do it. Okay? Okay, let's just come here. Do you have, somebody have a gloves? Could we borrow real quick? Thank you. So everybody just coming a little bit close. And also we have a Bunsen burner here. Do we have a Bunsen burner? Bunsen burner gave it to me. We need to move this a little bit because we need to use the rack of the sink. So move this around. Put it on the edge of. Do you have some? Do you have it? The places to do. Okay. First thing first. I have some new slides you can use, but you should have a box with some old one. Okay. First thing, I'm assuming it's all cleaned. Okay, you can clean it. We're gonna draw a big circle and write EC on the side, like this. Okay. Then we flip it over. After flip it over, we're gonna to turn on the Bunsen burner. Okay. At this moment, we want to try to test everything. Make sure this is working. Okay. Make sure the Bunsen burner is working. Do you have a hockey stick? Where is the hockey stick? Yes. Turn on the gas. Start to fire. This, the bottom one, is for you to adjust it. Okay, don't burn yourself. That's very important. Now we need a loop. Now all these loop looks like it's a little bit twisted. What you're going to do? Use hands to make sure they're straight. Because it's a soft metal. Okay, so what you do? Sterilize it. What do you mean sterilize? There's no microorganism, live body there, so make sure it's all red, a little bit on the top. So cool down a little bit. We're gonna add one big drop of the water right in the middle. Okay? Seems like there's a drop of the water there. That's good. Sterilize again. Because we want to carry on the bacteria, so sterilize it. Okay, now we have an E. coli growing there. That's good. So put this on the side, cool down a little bit, then we're going to pick the E. coli. Don't damage the agar, okay? Now we're going to make a big smear. A big smear like that. It's thin, and it is also like averagely covered the circle. Then go back to sterilize this. Make sure its bacteria is killed. Okay, now you put it back. Lots of these metal loop is twisted. You have to make sure they're straight. Now let's do the air dry. 3 to 5 minutes air dry. Now what we do? Let's say 3 to 5 minutes is ready. This is what you're gonna learn today. Our faucets to do the heat fixing. Back and forth couple times, but not cooking, okay? Not like stay in the middle and then chatting. Back and forth couple times. But you have to do this basically completely air dry. Otherwise you may see some legs. Back and forth. Okay, now ready. Make sure this is turned off. Because otherwise the fire is everywhere. That's just for the safety, very important. Okay, now what we're going to do. Come here to move right here on the stage. Okay, I think this is a little large, so make it a little small. Now what you need to do is a goggle. Very important. Before you touch any die, please wear a goggle. So you can wear a goggle like this. And the goggle, what we have to do is cover your whole eye. It's not the machine shopper using those big glasses. That's different. Okay, medical or experimental goggle is a little bit different. So what we're gonna do? Let's just choose something. Okay, saffroning. Let's pick saffroning, which is pink. So big drop in the middle. That's enough. Let's do staining for 1 minute. And let's say 1 minute is ready, and by this time you can prepare all these paper towel, just general paper towel. So let's say 1 minute is ready, so you use faucets, and very gentle, rinse, put it here, then we cover it. Now, ideally, this is already finished. This is already there. So, underneath microscope, we're going to do the observation, okay? So what we're going to do, we're going to try the microscope. I failed the last time, didn't show you very well. Let's do this time. So, show you one more time how to use the microscope. I hope this one works. So we'll see. It's already been 4 classes, 4 lab sections, so we'll see what it looks like. Again, let's show one more time. And in the lecture, we're going to mention again for the briefly for the light field microscope. Turn it on all the way to the top. Okay, we're going to use 10x, find the specimen, 100x with oil immersion. Make sure the oil is ready to see the morphology and the arrangement. So let's put the E. coli right there, right on the stage. See, this is a 2-side. Look at that, it's 2-side. Okay, so we gonna put 10x right there and make sure iris diagram is at 10. It's too shiny otherwise. So move all the way to the top, to the place you cannot move. Okay. Hope this works. Okay. It looks like there's a lots of bacteria there. Go ahead to see it. Usually at this stage is easy. 10x looks okay. Did you see?
Yeah.
Come here to see. That looks fine. See? You see a lot? Go ahead to see it. Yeah, that's what underneath the 10x. Lots of dots there. Okay, that means you find a specimen. Now, let's see whether we're successful. Okay, one twisted, second twisted, have an angle there. Then we're gonna put a big drop of the oil, a huge one, a big drop. Let's say whether we works. So we move here. Ideally, it's very tiny adjustment. We should see it. Seems like it's not. Maybe need to transfer another microscope. Let's try one more time. Go back. See whether it works. Okay, now I need to change the microscope. It's not work very well for this one. So we move on the side. Let's try this microscope. Because I did the last one, it works. So we'll see. We'll see to see whether it works. Okay. Most of the time I use crystal violet, but the first time we're using saffroning. So let's see whether it works. It happens lots of the time. It doesn't work very well. So we'll see. Goes a bit to the top. Again. And next. I'll show this one. Okay, I hope to see here whether we can find some. So we change to 10x. Okay, well, it seems like it works. Very lucky. You have to try a couple of times. Then make sure this is at 100x. So that's why I tell you, sometimes these are not working very well. Let's look at that. It's a very beautiful large pink color because it's saffron. Go ahead to see it. Can you see it? Large, single large, and the E color. Go ahead to see it. Otherwise, you think I'm just make a story. Maybe I didn't see anything. We have 3 evidence. Is that right? Let's go ahead to see it. This is underneath 100x what it looks like. And I hope you can have the same, you can make the same quality like what I did is good. And now what you do, you can clean up everything, finally don't go, and take out of your gloves, use your cell phone to take a picture. It will be a very nice, simple stain to see the E. coli, what it looks like. When we do the gram stain, next week, Tuesday, we will do E. coli and Micrococcus and you will see the titrate. So you can compare lots, single lots, and the titrate coccell. What's the difference between that? Then we will move on to endospore stain with bacillus. You will see another difference type of results. Okay? Very good. Thank you. Now go ahead to do.